Hello, I am Professor Sims, and this video is going to be about your organic molecules, carbohydrates, lipids, and protein. The learning objectives for this unit include identifying common elements and structures found in organic molecules, talking about isomerism, functional groups, the role of functional groups in synthesizing polymers. We'll give examples and describe the function of monosaccharides and polysaccharides within a cell, characteristics of lipids, triglycerides, and phospholipids, how phospholipids are used to construct biological membranes, structure and characteristics of amino acids and proteins, and finally the biosynthesis products that can be used to identify bacteria. The most abundant elements in cells and in general are hydrogen, carbon, oxygen, nitrogen, phosphorus, and sulfur. Carbon is the molecule of life, at least on Earth. I don't know how it is on any other planet, but on Earth, carbon-based beings are the life forms. And each carbon atom can bind to another one that produces a skeleton that can be straight or branched, or very often you see ring-shaped benzene molecules. Isomers, so they have the same atoms that are bound in a different sequence. That's what you call a structural isomer. Or if they have the same atoms, the same bonding sequence, but different spatial arrangements, so different shapes, those are stereoisomers. Now, functional group, this is what you call, if you hear somebody talking about a hydroxyl group or methyl group, aminophosphate group, those are what gives certain molecules very specific chemical properties, and we'll touch on some of those in a bit. Macromolecules are polymers that are bound together like building blocks. So macromolecules are polymers, and polymers are monomers that are bound together. Okay, I know, so much terminology in this. Some macromolecules are formed by what is known as dehydration synthesis, and this is a reaction that occurs when monomers combine their functional groups, and a byproduct of that reaction is water. Okay, so let's look at some figures. These are some basic illustrations of common molecules. So you have your carbon dioxide. This would be an illustrated model of carbon dioxide with a carbon in the middle and two oxygens that are double bound. This here is ammonia. So there's nitrogen in the middle, hydrogen, 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 and then oxygen is just two oxygen atoms that are bound with a double bond. These are some examples of structural isomers. So what that means is they have all of the same atoms, but not all of the atoms are bound in the same sequence. So, and it highlights the differences. So you have glucose. Glucose is the simplest sugar. And then you have galactose and fructose. And all of them have the same chemical formula, C6H12O6, but they're not bound in the same sequence. So those are structural isomers. I've included this figure here in order to give a brief overview of all of the organic molecules that we'll be discussing. These are all macromolecules, which are rather large molecules. The four major macromolecules are your carbohydrates, lipids, nucleic acids, and proteins. The carbohydrates are the most abundant macromolecules. They're the source of immediate energy needed in living systems. They participate in defining the structure of cells in living systems. They include single sugars or multiple sugar molecules bound together into polymers. And collectively, there are three types of carbohydrates, which are the monosaccharides, disaccharides, and polysaccharides. So that's the carbohydrates. Next, briefly discussing proteins. These are polymers made of amino acids. And amino acids are small molecules that can contain amine, a carboxyl acid, and a side chain group. Amino acids are connected by peptide bonds to form protein polymers, and this gives rise to different levels of structure for proteins. Primary structure of a protein is made up of a string of connected amino acids. Secondary structure of a protein is formed by the coiling and folding of the primary structure. The tertiary structure of a protein is formed by interactions between the components of the secondary structure. And some proteins have a quaternary structure, which includes assembly of multiple individual subunits to form a functional protein. So we will cover that in much more detail in the later slides. Now, briefly, nucleic acids are the molecules inside the cell that store and process genetic or hereditary information. The two types of nucleic acid are deoxyribonucleic acid, which is in DNA, and then ribonucleic acid, which is found in RNA. These macromolecules are called nucleic acids because they were first found inside the nucleus of a cell, and the monomers that make up nucleic acids are called nucleotides. Each nucleotide contains a sugar, a base, and a phosphate group. Finally, the lipids, briefly. Lipids are macromolecules that are all insoluble to water. That means they cannot dissolve in water. They include your oils and fats, phospholipids, and steroids. Oils are found in plants, where they're used for long-term energy storage. Fats are found in animals, where they also provide long-term energy storage as well as insulation. Fats and oils are composed of glycerol and fatty acids. A fatty acid is a long chain of carbon-hydrogen bonds with a carboxyl group at one end. Based on the number of carbons and the number of double bonds that are present, fatty acids can be classified as saturated or unsaturated. Again, we'll cover this some more in the later slides. Fats and oils are formed in a dehydration reaction in which 3 fatty acids react with the OH group in glycerol. Dehydration synthesis, and you see how it also has a byproduct of water here. So this is 2 molecules of glucose that link together, and this product is maltose, which is a larger sugar, and then of course water was produced in that reaction as well. Carbohydrates are the most abundant biomolecules on Earth. Carbs are used not only for energy, which we already know, but for structural purposes in cells. Carbohydrates are essentially sugar molecules that are linked together. So a sugar molecule is called a monosaccharide. Monosaccharides link together via glycosidic bonds, and then they have classifications that are based upon the number of carbons that they contain. So if it has 3 carbons, that's a triose, 4 carbons is a tetrose, 5 carbon pentose, 6 carbons hexose. And anytime you hear something that ends with ose, they're talking about carbohydrates and or sugars. Monosaccharides are building blocks for polymers and complex carbohydrates. So here we have monosaccharides, aldose, ketose, triose, pentose, hexose, right? That's their classification based on the number of carbons that they have. And then this is glyceraldehyde, dihydroxyacetone, glyceraldehyde, ribose, glucose. So it has to do with how many carbons it has, and then the position of the carbonyl group. Sucrose, lactose, and maltose. And these are composed of 2 monosaccharides that are linked together, again by glycosidic bonds. Polysaccharides are polymers that have hundreds of monosaccharides. So these are very, very, very big carbohydrates. Starch and glycogen are examples of polysaccharides. In the lab, we're going to be dealing with, we're going to be working with all of these, well, not all of them, but all different sizes of sugars and carbohydrates. So we will be working with glucose, which is the smallest sugar, and then we'll have monosaccharides. Then we're going to go up into disaccharides when we're working with sucrose and lactose, and then we'll be dealing with starch hydrolysis. So we'll be dealing with polysaccharides at that stage. And those are basically branched, really, really long chains of glucose. Cellulose is a structural component of cell walls. The neat thing about cellulose is it actually can't be digested by humans. It can't be digested by a lot of things. Cellulose is commonly found in things like paper, right, and wood.
And that's how come things like termites can just destroy homes and trees like nobody's business, because they found a niche. They can eat the cellulose, they can digest it, and basically nothing else can. So they can hide up in there away from any kind of predators and have all of the food to their heart's content. But as far as cells are concerned, yeah, cellulose, it's very dense and thick, and it is large, and it provides a lot of structure to cell walls. Other structural polysaccharides, you've got N-acetylglucosamine, N-acetylmuramic acid. These are found primarily in peptidoglycan and chitin. So you see a lot of these polysaccharides making up the cell envelopes of especially gram-positive bacteria. So these are some of the most common disaccharides we talked about already. We've got maltose, right, and lactose and sucrose. See how they are similar in structure, and they call them disaccharides because they are linked monosaccharides, and what links them here is a peptide bond, right? Here is some starch and glycogen and cellulose. So these are their structural diagrams, like their chemical diagrams, and then these are some electron micrographs of starch, glycogen, and cellulose. Cellulose is very fibrous. It's very tough. Lipids are mainly carbon and hydrogen, but they also can contain oxygen, nitrogen, sulfur, phosphorus. So lipids, they provide nutritional value. They consist of carbon and energy stores. They do provide some structure. They also can be hormones. They're used in pharmaceuticals, fragrances, and in pigments. Lipids they're composed of fatty acids. Fatty acids are long-chain hydrocarbons with a carboxyl acid functional group, usually at a very specific location. Fatty acids have long non-polar hydrocarbon chains, which means they're hydrophobic, so they repel water. You've seen this. When you put oil in water and they don't mix, it's because lipids are hydrophobic. Fatty acids that do not have double bonds are saturated. So you've heard of saturated fats. Those are ones that do not have any double bonds. And unsaturated means that they do have double bonds. Fatty acids chemically bond to glycerol to form triglycerides and phospholipids. So this is figure 7.13. It shows a phospholipid with two different fatty acid chains. The one on the left is the saturated fatty acid, and the one on the right is unsaturated, and you can tell the difference because this one only has single bonds and this one has a double bond. Triglycerides comprise three fatty acids that are bound to glycerol, and they are hydrophobic as well. Phospholipids contain a hydrophobic hydrocarbon chain and polar head groups, which makes them really large-scale structures. So we'll look at that in a bit. Membranes, biological membranes, cell membranes, organelle membranes, are based on this phospholipid bilayer, where they have hydrophilic exterior and interior surfaces. So you want to have the transfer of liquids and things like that. So on the inside and on the outside, it's hydrophilic, which is it's good for if you live in an aquatic environment, which most cells generally do. Especially for organismal cells. But then they have to have this hydrophobic layer in order to keep the liquid in, the cytosol in, and keep themselves separated from their aqueous environment. The bilayers are the structural basis for cell membranes, subcellular components. Isoprenoids are lipids that are derived from isoprene that have many physiological roles in commercial applications. So let's have a look at, this is figure 7.12, triglycerides. So triglyceride is a glycerol molecule that has three fatty acids connected to it. And this does occur via dehydration synthesis reaction. So again, see, we have a byproduct of a triglyceride being formed is water molecules, right? So here's your glycerol, and then your three fatty acid chains are added, and then the hydrogens from the glycerol and the hydroxyl groups, the functional hydroxyl groups from the fatty acid chains, combine to form water as the byproduct. These are phospholipid bilayer, so they tend to arrange themselves in aqueous solution, forming lipid bilayer sheets. You have nonpolar tails and polar heads. So you have your hydrophilic layers on the outside and on the inside. Hydrophilic means water-loving, and then hydrophobic layer in the middle. Wax is a long-chain isoprenoid that is very water-resistant. An example is sebum. This is produced by your sebaceous gland, your oil glands in your skin. So those are actually wax isoprenoids. Steroids are lipids that have ringed structures that function as structural components of cell membranes, and also, of course, steroids are hormones. Sterols are a subclass of steroids that contain hydroxyl group at one end of the ring. An example of a sterol is cholesterol. Hopanoids are structurally similar to cholesterol, and they strengthen bacterial membranes. And then, of course, you have fungi, protozoa. Those guys have ergosterol. We talked about that when we talked about fungi. Here's some more chemical formulas. These are 5-carbon isoprene molecules that have been modified in different ways to yield isoprenoids. This here, you guys are familiar with beta carotene, the stuff that's in carrots. Let's have a look at amino acids. So amino acids are the building blocks of proteins. Each amino acid has an alpha carbon that is attached to a hydrogen atom and a carboxyl group and an amine group. There's a 4th group in each amino acid, and you usually see it written as the letter R, and that has to do with different groups. So R represents a different group, usually that contains carbons, and they have different chemical composition, size, polarity, or charge, which gives the different amino acids their varying properties. And then, of course, peptides are polymers formed by the linkage of amino acids, very similar to the linkage that we saw earlier in monosaccharides to form disaccharides or polysaccharides. So very similar. These are, this is figure 717, and these are your most common amino acids, your lysine, glutamine, aspartate, serine, cysteine, alanine, right? So you will sometimes see these written as R. Check it out. If you were to delete, just cover up and delete everything that's in a blue box, okay? What you will have is a molecule with exactly the same structure, right? It's the stuff that's in the blue box, what they call the R group, that gives these different amino acids their different properties. And then, of course, the bonds between different amino acids that are linked are peptide bonds. And then it's not showing it. Oh, yeah, it is. Look. So this, again, is a dehydration synthesis reaction, and you can tell because it has a byproduct of water, right? Very similar to what we saw with the monosaccharides. Proteins are polymers that are formed by linking, it says several amino acids. It's really, usually it's a very, very long chain of amino acids. Proteins are nutrients, they are enzymes, they are storage molecules for carbon, nitrogen, and energy, and they give structure to cells. So proteins are very important. Structure, remember this, structure determines function in proteins. And there are 4 classifications of protein structures, which are primary, secondary, tertiary, and quaternary. And the native structure of a protein can be disrupted by denaturation, and this makes the protein lose its structure and function, right? Because structure determines function. So if it loses its structure, it also loses its function. Some proteins are formed by subunits, interaction of which compose quaternary structure of protein complex. I'm going to try to explain this with a figure in just a moment. Conjugated proteins have non-polypeptide portions that are either carbohydrate, if it's a carbohydrate in a conjugated protein. We know that the polypeptides is how amino acids with peptide bonds. Okay, so I don't confuse you, just remember conjugated, a protein that is conjugated with a carbohydrate, that's forming a glycoprotein. A protein that is conjugated with a lipid, a lipoprotein. These are also important components of membranes, cell membranes, organelle membranes. So the primary structure of protein is its sequence of amino acids. And like I said, it's usually a very, very long sequence of linked amino acids. Okay. This is figure 723. So this is showing primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure, like we saw already, is this very long linked up chain of amino acids, just like a string of pearls or Mardi Gras beads, okay? The secondary structure is how the proteins are folding. So you have folds of polypeptide chains, and they either turn into sheets or helix, right? Then tertiary structure, this is like a 3D folding pattern of a protein that has to do with side chain interactions. So proteins get very long and they're complex, and then you have these little side chain interactions with other proteins and other things. So this is where the tertiary structure comes in. And then quaternary structure has to do with more than one amino acid chain, okay? So you've got the amino acids and you've got these foldings, and then you have these side chain interactions, and then if you have a whole nother amino acid chain attached to it, then you're dealing with a quaternary. So this is figure 722. This is a hemoglobin molecule that is showing two alpha polypeptides that are bound together with two beta polypeptides and 4 heme groups. So this is just showing just how complex and rather large protein molecules can be, which makes sense since proteins are so integral in as structural components of cells. So section 7.5 is talking about biochemistry as it relates to identifying microorganisms. So of course, identifying bacteria is essential for diagnostic purposes, also for research purposes, and for managing outbreaks, epidemics, pandemics, dealing with food poisoning. Those are all examples of reasons that you would need to identify bacteria. Phenotypic identification involves observable traits, so things that you can see. And this has to do with structural components, for example, lipids, different proteins, enzymes, biosynthetic products like sugars, amino acids, storage compounds like polybeta hydroxybutyrate. Say that one 3 times fast. An unknown microbe can be identified from a mass spectrum produced when analyzed by matrix-assisted laser desorption ionization time of flight mass spectrum. MALDI-TOF is what they call it. Sounds a little bit like Molotov, right? Well, this is what it looks like. This is figure 726, MALDI-TOF. This is used for routine diagnostic procedures in a clinical microbiology lab. It's able to rapidly identify some microorganisms that can't be identified using more traditional methods. Now, more traditional methods would be things like gram staining, testing for the presence of certain enzymes. We're going to dive into that quite a bit in the lab. Okay, but for MALDI-TOF, essentially what you do is you grow the microbes out on a special plate, you expose it to irradiation, and then you use a mass spectrometer to measure ions that are released after it's exposed to irradiation. And then you can compare the ions that are released to profiles of known species. And then that way you can identify the unknown species. You can also use lipid composition and fatty acid methyl esters, which is also known as FAME, or phospholipid-derived fatty acids, PLFA analysis. You can do proteomic analysis. This is where you're looking at all of the proteins in an organism. You can use glycoproteins in the plasma membrane or in the cell wall structures. And then, of course, like I mentioned before, we're going to use more traditional micro and biochem methods when we do our semester project in the lab. We're going to go into that in great detail. In fact, starting with Lab 6 and going all the way through the end of the semester, we will be working on our unknown projects. And it starts with very traditional techniques like gram staining, endospore staining, acid-fast staining, and then we'll test for things like, catalase, enzymes, coagulase, all that fun stuff. This is figure 727, fatty acid methyl ester analysis. So essentially all you do is you grow out your culture, and then fatty acids are extracted from the bacteria and they are converted to methyl esters. And then you use a gas chromatogram to analyze the methyl ester fingerprint. And then again, you can compare the fingerprint that you get from the unknown to those of already known species.
That is the conclusion of the Lesson 6 material. As always, thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and then leave your questions for me in the comments section below.